we need to understand this biological weapon, gain of function a viron that was basically made in a lab. And we need to understand how it interferes with our immune system and how it basically creates this inflammatory process. And once we understand that, then we can understand why is it that a lot of the healthcare practitioners and in the alternative realm, and of course also the mainstream medical conventional doctors that now are recognizing the value of some of these supplements, etc. And so to understand the virus, whether you want to call it a bioweapon, lab made, whatever it is, you have to look at its characteristics. So it's very, moderately small in size. So depending on what literature you look at, it'll say it'll be from like 50 nanometer to 90 nanometer, which is moderately small. It is a single-stranded mRNA virus, so it's not a DNA virus. It's an RNA virus. I'm sorry, that's a typo. It's an RNA virus, not mRNA. RNA virus, single-stranded, and the RNA, which is the genetic material, is inside of the cell, which is enveloped by a lipid cell wall, the lipid cell wall, which is its Achilles tendon, because you can wash lipid with soap and it will dissolve. And then you have the spike proteins, or S1 spike proteins, which are responsible in the binding to a human cell receptor that is called ACE2 receptor. And it is moderately virulent and infectious. And it's not, let's say, as severe as something like Ebola, for example, or Zika virus. It's moderate in that infectivity and virulent. And also, it uses a protein enzyme, the TMPRSS2, and this is what activates the lock and key mechanism of the spike protein with the human ACE2 receptor site. The reason I'm mentioning these, because these are the Achilles tendon that we will see how we can neutralize when we're dealing with COVID long haul, even the vaccine side effects. Because remember, the vaccine is injecting an mRNA, genetic material inside of your cell that hijacks the factory of the cell, just like COVID does. So just like the virus does that by inserting its mRNA into the cell, hijacking the factory of the cell, asking the ribosomes to produce the spike protein in your cells. So both the virus does that, but also the vaccine, the mRNA vaccines do that, as well as the other attenuated viruses in AstraZeneca and other vaccines. And so the spike protein attaches to the ACE2 receptor site. And if you look at this slide, you will see that, so the ACE2 receptor is on the cell wall of the human cell. And then you have a protein serine enzyme that is TMPRSS2 that activates this lock and key mechanism. So the spike protein latches onto the ACE2 receptor with the enzymatic catalyst that makes this process happen, which is this protein. And so once that lock and key is established between the virus and the healthy cell, then the virus gains entry into your cell. It gains entry to your cell. Once it gains entry to the cell, it injects its RNA, single-strand RNA, genetic information inside of the cell. And once it shoots that RNA information into the cell, then your cell factory is hijacked, where mRNA, a messenger RNA, is carrying the code, the genetic code, to tell your cell factory to start replicating the spike protein. If it is the mRNA of the vaccine, it does the same thing. It injects the mRNA of the vaccine into the cell, and it asks the factory of the cell, or commands the cell, to produce the spike proteins. In the case of the virus, once it hijacks your factory of the cell, it produces the spike protein, but the whole factory of the cell also starts to replicate the RNA and puts together new virons, the new viruses that now incubate in your system to a number that can cause infection if your first line of defense does not come in and block that replication from happening. So the first line of defense in the immune system, which is basically the presence of macrophages, natural T killer cells, monocytes, neutrophils, mast cells, eosinophils, basophils. These will encounter the virus in your nasal passages, in your trachea, in your nasopharynx, by your ciliated cells and goblet cells, in those nasal passages, in the airway system, on contact. And your first line of immunity will kill right on the spot the virus, whether it's a natural occurring or this biological, gain of function research virus. And on the spot, it would neutralize and kill them. This is your first line of defense. If this happens, then you're successful. You don't even go to the second line of defense, where which is called adaptive immunity, where you need to make antibodies. The first line of defense is called the innate immunity. Through some of these immune cells, the white blood cells, etc., it doesn't even allow the virus to replicate to a number that then your second line of defense, T and B lymphocytes, come in and make the antibodies. And if your first line of defense is unsuccessful and this gets into the lower respiratory system, the virus replicates and gets down to the deeper lungs tissue. It passes the bronchioles, it passes all of that, and it goes to the alveoli of the lung tissue. And once it starts to hijack those cells and starts to replicate itself, then this is where you get the cytokine storm, where all of these first line of defense immune cells, even though they weren't able to neutralize the virus from replicating or attaching to more and more and more ACE2 receptor sites, you get a barrage of macrophages, T helper cells that basically will send an alert, help, 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 to bring in the inflammasomes, the cytokines, which are a cascade of inflammatory cells, interleukins, NF-kappa Bs, etc., that create the problem of acute respiratory distress syndrome or disease, etc., through the cytokines. Once you have the cytokines coming in through all these inflammatory cells, then you have what we know, platelet aggregation, you have blood clots forming. Of course, the endothelial cell linings are involved when they start to replicate the spike protein, which is also the reason for myocarditis, stroke, whether you had COVID last year in hospitals, you heard people are having strokes, heart attacks, etc., kidney failures, etc. Well, some of it was because of remdesivir, but a part of it has to do with the blood clot forming and the endothelial cell linings being impacted, and the platelet aggregation takes place, blood clot formation, and then there is a process of tissue repair by the means of fibroblasts that come in to do the tissue repair, which also triggers certain other inflammasomes such as NLRP3, which is a cytotoxic for tissue death. When a tissue is damaged, that cascade of cells come in to basically get the dead cells out, etc., which causes the lungs tissue to become fibrotic, and other tissues, also tissue necrosis takes place, and that is a problem. We don't want to go there. So all of these little names I throw at you, NLRP3, etc., ACE2 receptor, the protease, serine protease, that is the enzymatic catalyst to have that lock and key mechanism, is because what we're going to see next. So if your first line of defense is not successful, if you don't make the antibodies in time, which is the second line of defense, to stop the cytokine storm from happening, or have it in a mild fashion, not to the extreme where your lungs tissues are incapable of doing their work, etc. So remember these: NLRP3, ACE2 receptor, TMPRSS2. So there is a reason why I want you to remember that.